Hello, welcome to your own channel Curious Corners in Biology, where biology is made interesting because the scientific approach to the subject is discussed. This is all about hows, ifs, buts and whens of the subject. So today we are going to discuss two interesting questions. The first question is that what is considered as living and non-living in a cell? What is the criteria to determine whether something is living or non-living in a cell? Okay. So, something without which a cell cannot survive, a cell cannot exist, is considered as living. Structures which are actively or directly associated with the metabolism of the cell, which help the cell to respond to the environment, are also considered to be living. For example, cell membrane, ribosomes, lysosomes, nucleus of the cell. These are the structures without which a cell cannot survive. Okay. So, these are living structures. Of course, these structures do apply to eukaryotic cell only, but there are exceptions to this. For example, there are questions like what about RBCs and sieve tube elements? These cells are enucleated. RBCs do not even have cell organelles. Then how are they considered to be living? Because yes, they are considered to be living cells. The answer to this is that these cells are highly differentiated cells, and the cells from which they are formed did not have they were not of the constitution that they did not have nucleus. They did have nucleus, they did have all the cell organelles, but because they differentiated into specialized cells, say for example RBC has got a very specialized function of transport of respiratory gases, for which it needs to pack maximum amount of hemoglobin into its volume. So, it cannot simply afford to have nucleus within it. And remember, not all RBCs of animals are enucleated. Of course, there are lower vertebrates, of course, there are mammals as well whose RBCs are nucleated like camel. But for humans, the energy requirement is so high that the hemoglobin needs to be packed in maximum amount. Having nucleus in the volume of the cell simply will not allow the amount of respiratory gases to be transported which a body needs. So, hemoglobin has to be packed full inside a mammalian RBC. Now, if we talk about sieve tube elements, then sieve tube elements are cells of phloem of the plant, and they do have a function of transport of organic food along with water, right? Now, this transport is called as translocation, and of course, sieve tube elements, when they pile upon each other, they will form a passage which is called as sieve tube. Now, this passage is a freeway. That means anything should not obstruct the transport of organic food and water. So, nucleus should also not come in the way. So, in the process of differentiation, they have lost nucleus. So, this is the answer as to why living structures are not present in certain highly differentiated cells. What about non-living structures in a cell? How can we call certain structure or component non-living in a cell? So, anything without which cell can survive is simply considered as non-living. This non-living components do not actively participate in the metabolism of the cell. They are not actively involved in assisting the cell about how it responds to the environment. Say for example, cell wall. Cell wall is a rigid and stiff non-living structure. You might have read this. Cell wall is not actively involved in metabolism. It does not directly respond to any environmental stimulus. And cell, whether bacterial cell, plant cell or a fungal cell, if it has its cell wall removed, then cell can still survive. So cell wall by itself is non-living. Same applies for cell inclusions as well. Cell inclusions are pigments in the cell, crystals, reserve food material. All these things, if they are removed from the cell, again the cell can survive. So cell inclusions are also non-living. So here we understood that what is the criteria for living and non-living. So if you like such content, please do not forget to hit the like button and please subscribe to my channel. Okay. The second question which we are going to discuss is mesosome an artifact? What do we mean by artifact? Artifact are structures which are not naturally existing for a cell. So does that mean that mesosome does not exist in a bacterial cell? The answer is yes. Okay. Up to 1960s, it was thought that bacterial cell membrane formed infoldings or invaginations of the cell which are called as mesosomes. Now mesosomes had functions like cellular respiration. It had all the enzymes for cellular respiration. It housed all such enzymes for breaking down of food. Then it did play a role in distribution of genetic material during binary fission. It also had a role in cell wall synthesis. But in 1960, it was found out that mesosomes are not actually existing. Mesosomes were observed in a bacterial cell as a result of chemical fixative techniques or chemical fixation techniques employed in electron microscopy. Chemical fixation is done to make any specimen stable, and usually chemical fixation in electron microscopy is done by using aldehydes like formaldehyde, glutaraldehyde. These aldehydes are known to link proteins and lipids. So probably by linking the proteins and lipids of cell membrane, these infoldings or invaginations might have been formed, which were thought as mesosomes. To the extent that there was a functional comparison of mesosome to mitochondria as well. But then when using other techniques for preparation of specimen for electron microscopy like freezing techniques, mesosomes were no longer found. So mesosomes were established as artifacts of the cell. These are not naturally occurring structures. They are no more believed to be existing for a bacterial cell. Okay. So that much for today. As you know, this channel is based on my book Curious Corners in Biology, which is given in the description. The link of the book is given in the description. You can visit the link, read the description. Okay. Thank you very much. See you next time. Bye.